presentation my research title, Characterization of New Molecules Inhibitor Effect on Alzheimer's Disease Amyloid-Beta Peptide Cascades. My presentation containing three main topics: Alzheimer's disease history and mechanism of plaque and tangle formations, and discovery, our new discovery molecules effect on the amyloid cascades such as A-beta peptide aggregation, A-beta peptide neurotoxicity, and memory impairment. In the year of 1901, August Dieter, a 50 years old woman, was admitted to the hospital because she was under the weather for a long time. Her family reported that she has strong feeling of jealous and agitation and mood swing. He is the Dr. Alois Alzheimer, German neuropsychologist. He found that he inspecting her health during the hospital day. He found that she read the newspaper but did not seem to understand, and she has facing a lot of difficulty to carry out the regular task. And in additionally, unfortunately, in the year of 1906, August Dieter was died. Dr. Alois Alzheimer found that some unusual features present in her brain after postmortem report. Actually, he observed that the plaques and tangles spread throughout the brain. This is a main reason her brain shrunk drastically compared to the normal health brains. The disease bear his name after he died. Alzheimer's disease is the most prevalent dementia among senior populations. It succumbed to around 45 million people and forecasted 150 million people can reach by 2050 unless we predict effective treatments. Every 3 seconds, someone has developed the Alzheimer's disease. Let's see what is the plaques or tangles. The amyloid-beta proteins aggregated around the neurons is called the amyloid plaques. Then hyperphosphorylated tau proteins accumulated inside the neurons, which called the neurofibrillary tangles. The neurofibrillary tangles and plaques play a significant role to destroying and disturbing in the neurons. Okay. Actually, amyloid precursor protein genes has three variants: APP 695, APP 751, and APP 770, which are produced by neurons, endothelium cells, and platelets. In generally, the neuron, the amyloid-beta peptide aggregation characterized by the two ways. One is the amyloidogenic pathway, another is the non-amyloidogenic pathway. In the case of amyloidogenic pathway, the amyloid precursor protein, which contains 695 amino acids. The amyloid-beta proteins represented by the green, which release from the amyloid precursor protein after cleavage by the beta secretases within the membrane and gamma secretases in the extracellular membranes. In the case of healthy brain, the A-beta peptide exists in the monomer state with soluble natures. These proteins are main responsible for neuronal development, including productive neurons from the cell toxicity. In the case of Alzheimer's disease patient brain, the A-beta peptides tend to form A-beta dimer and trimer, tetramer, and soluble oligomers, and finally transform into the insoluble fibrils. The fibril converted into the plaques. The soluble fibrils can reach 3 to 5 kilodalton. It can disturb the mitochondrial part. then it can increase in the reactive oxygen species. In case the soluble oligomers can reach 10 to 100 kilodalton, it exerts more cytotoxicity compared to the amyloid-beta fibrils. A healthy brain has got 100 billion neurons, each with a long, the branches extensions. These extensions forming connection with another neurons. This connection called synaptic cleft. In generally, the information transported as a tiny pulsed chemical released from one neurons and taken up by the another neurons. The brain containing 100 trillion synapses that are allowed signal to travel rapidly through brains. This signal create cellular bases such as memory, sensation, emotion, and skills, cognitive skills. In the case of Alzheimer's disease patient brain, the A-beta peptide, the amyloid plaques and neurofibrillary tangles can damage the neurons as well as killing the neurons, which can causes to damage, causes to cognitive impairment. So far scientists are targeting amyloid beta plaques to cure the Alzheimer's disease. Although 100 clinical trials tested in the clinical test, recently on June 2021, FDA licensed aducanumab as a drug molecule for Alzheimer's disease patient treatment. But unfortunately, the Committee for Medical Product for Human Use rejected to use aducanumab because it cannot alter the course of Alzheimer's disease. So that now there's only one option. There's one promising options to cure the Alzheimer's disease. We have targeting amyloid A-beta peptide oligomer, reducing the A-beta peptide oligomer toxicity by interacting with the effective drug molecules. In this perspective, we can identify effective small molecules. Actually, we employed Schrodinger tools to identify the molecule M30 from 13.2 million drug-like molecules, which is deposited in the Jing database. Okay, by testing the Lifkin's rules, Jarkin's rules, and blood-brain barriers, and other pharmaceutical properties such as human oral absorption and central nervous systems. In addition, we can examine the peptide, the A-beta peptide secondary structure changes in the presence and absence of M30 molecules. The mostly accepted hypothesis that the beta seed formation in the amyloid beta proteins is a key event to increase in the aggregation of A-beta proteins. So we found that in the presence of M30 molecules, the A-beta peptide the A-beta peptide containing the beta seed has been reduced significantly compared to the wild type A-beta proteins. So the M30 molecule can decrease in the beta seeds. And in addition, we examined the A-beta peptide aggregation in the presence and absence of M30 molecules after incubator of 37 degrees for 16 hours by using the 16 hours. Okay, this figure showing that the A-beta peptide aggregations found to be higher in the case of A-beta peptide, by indicating by the red lines. Whereas in the case of A-beta M30, the aggregations of A-beta peptide can be degrees, which is represented by the blue color lines. So this data imply that M30 molecules can decrease in the A-beta peptide aggregations. Furthermore, we use confocal microscopy to inspect the A-beta peptide accumulation on the new PC12 neuronal cell membranes. As shown in the figure, the first case is the A-beta peptide, the second case the A-beta M30 A peptide. The neurons are identified by neurons can see in the red lines, and A-beta peptides are represented by green fluorescence. At the bottom of the snapshot, we can see the green light, the green fluorescence can deposit over the neurons. These depositions has significantly reduced by the M30 molecules. So this evidence can imply that A-beta peptide associated on the neuron member has been reduced significantly in the presence of M30 molecules. A mostly accepted hypothesis that the A-beta peptide can interact with the membranes and generating the ion channels causes to the calcium ions flows increases in and out of the neural membranes. It can generate in the new reactive oxygen species as well as the damage the neurons. So it leads to killing. It leads to neuron death. So we examined the calcium ions flows on the PC12 membranes in the case of A-beta and A-beta M30 molecules by using the fluorescence spectroscopy. This data represent that the calcium flows was increased. The higher amount of calcium flows we observed in the red lines. When we interacted with the M30 molecules, the calcium ions flow in and out of the neural membrane has suppressed significantly. So this data revealed that M30 molecules can decreasing the calcium ions flows. Okay. Then then we have to examine A-beta peptides presence in the neural membranes. It carried out the in vivo studies. Actually, we take out the brains from the A-beta from the mouse. After A-beta and A-beta M30 injecting to the mice for long times. Actually, the top snapshot showing that the A-beta injecting to the mice for 2 weeks. The bottom snapshot shows that the A-beta M30 molecules interacting into the mice for long times. The green fluorescence represent the A-beta peptides. We can see in the merged figures the green intensity is higher in the case of mice treated with the A-beta proteins. This green intensity has been disappeared in the case of A-beta M30 molecules, but this treatment carried out for several months. So these data are imply that M30 molecules can removing the A-beta peptides. Prior to removing the brains from the mice, we can examine and learning and memory test of the mice by making the several trials. The left side of the video, shows that A-beta injecting to the mice for 2 weeks. Within the 2 weeks, the A-beta peptides can alleviating the neuronal cell can increase in the neuronal cell death, which causes to memory impairment. So this mice can take a long time to figure out the exit point. On the other case, the A-beta M30 molecules injected into the mice for several months. After that, we examine the learning and memory test. These mice can reach the exit point quickly. So these data are strongly supported that the A-beta peptides, the M30 molecules improve the memory of the mice by decreasing the cell death and A-beta peptide association on the membranes. In a nutshell, we recommended the M30 molecules to use the clinical trial since it can block multiple steps of amyloid cascade, such as A-beta peptide aggregation, A-beta peptide association on the neural membrane, and stop the pore formations on the membranes. And then the in vivo studies, the M30 molecules improve the spatial memory by destroying the A-beta aggregation and reducing the neural cell death. Okay, the following funding agency assist me to carry out the project on Alzheimer's disease with those mentors. Thanks for your attention.